Good morning, children. Let us start the next topic, Mendelism. The contribution of Mendel to genetics is called Mendelism. It includes all concepts brought out by Mendel through his original research on plant hybridization. Mendelian genetic concepts are basic to modern genetics. Therefore, Mendel is called as father of genetics. Father of genetics, Graham Johann Mendel. Life history of Graham Johann Mendel is given in page 34. He was born on 22nd July 1822 in Austria. Was also a mathematician, philosophist from University of Vienna. He worked as a priest at Augustinian St. Thomas Monastery. He worked as a secondary school teacher. From 1856 to 1863, he did his historically important hybridization experiments on pea plant. Continuously for 7 years, he did his experiment and recorded his data. He presented his paper entitled as Experiments on Plant Hybrids. It was published in Proceedings of the Bern Society of Natural History in 1866. Mendel was the first systematic researcher in the field of genetics. So he was celebrated as father of genetics. So important question as well, you will get 1 words and 2 marks from this passage. Please read it carefully. Mark and learn all the important 1 words. Next we are going to see about why pea plant. Why he has selected pea plant for his studies. Because it is an annual plant. Also it has clear contrasting characters that are controlled by a single gene separately. Next point, self-fertilization occurred under normal condition in garden pea plant. You know what is self-fertilization. Mendel used both self-fertilization and cross-fertilization. In his experiment, he followed both kind of fertilization. For doing cross-fertilization, it has to be done, that is, the pollination has to be done manually. For that, the floral part should be large enough. In pea plant, the flowers are large sized, so it is very easy for doing this hybridization. Because of all the above 4 reasons only, he has selected pea plant for his studies. What are the 7 different traits? So when we are seeing the traits, there is seed shape. For example, seed shape is a character, means there is a dominant trait and recessive trait. As well, there are 7 different traits. There are 7 different characteristic features. All the 7 different characteristic features shows dominant and recessive characters. Seed shape, dominant, recessive. Round, dominant, wrinkle, recessive. Seed color, yellow, green. Flower color, purple, white. Flower position, axial, terminal. Plant height, tall, short. Pot shape, inflated and restricted one is constricted. See, the recessive trait is constricted. You can able to see the structure. Inflated means without any division or without any partition. In constricted, you can able to see the partitions as like waved outer line, isn't it? So that is constricted pod shape. Next one, pot color. Green color is a dominant one, yellow color is a recessive color. Now the next one, Mendel was successful. Why? What is the reason? Because of 5 important reasons. One is he applied mathematics and statistical method to biology and laws of probability to his experiments. Based on this, based on mathematics and statistical method, he recorded his experimental results. And he followed scientific method and kept accurate detailed record that include quantitative data of the outcome of his cross. What is the meaning quantitative data? Even the numbers. For example, one pea plant, nearly many seeds will be produced, isn't it? All the seeds, from all the seeds, he has taken the sapling and he has counted the each and every sapling and what is the character of this each and every sapling. So, and likewise, he has recorded. So that is what said to be as quantitative data, right? He followed scientific method and kept accurate and detailed records that include quantitative data of the outcome of his crosses. His experiments were carefully planned and he used large samples. Okay, huge number, large samples. The pairs of contrasting characters which were controlled by factor were present on separate chromosome. Here, the genes are responsible for the characteristic feature. So each and every gene, okay. So the particular gene is present in a separate chromosome. 7 different characters he has taken. 7 different characters are controlled by 7 different genes. 7 genes are present in 7 different separate chromosomes. Okay, and then the parents selected by Mendel were pure breed lines, and the purity was tested by self-crossing the progeny for many generations. So, parents selected Mendel were pure breed lines. What are pure breed lines means? Generation after generation, the same parental character is carried over to the next generation, means it is said to be as pure line breeding. So, what is that? We will see in the definition part. Okay. So, because of all these reasons, Mendel was successful in his experiment. Okay, now it's a picture which denotes the 7 different genes which are located in different places. Okay, flower color, seed color, pod shape, flower position, plant height, pod color, and seed shape. So, location of genes for the 7 characters on the chromosome of pea plant. So, the location, the place where this gene is located, okay, that location is called as locus. That also we will see in the definition part. Locus is the place where the genes are located in the chromosome. So when this Graham Johan did all his experiment and recorded all his experiment, at that time the cellular biology or the genes about the studies about the genes or chromosomes were not yet developed. Okay, but still he has recorded all his experimental results. So his results were rediscovered by 3 different biologists. Okay, Hugo de Veres, Carl Eric Correns, Eric von Tuchemark. So these 3 biologists, they have rediscovered Mendel's publication, and then only his concepts started. It was started to accepted by all over, and then it started to practice. Okay. So this 3 persons name, it is very important in page number 37, first paragraph it is given. Please mark and learn. And in page 36, there are 2 different tables are there. What are the dominant trait? What are the recessive traits are given? Okay, so mark and learn the table 2. Now we are going to see about what are the terms related with this genetics. Now we will see about some terms and definitions which are related with genetics. Okay, first one is self-fertilization. You know what is self-fertilization? The fusion of pollen and egg of the same plant is called self-fertilization. So then what is cross-fertilization? The fusion of pollen egg of the different plant is fusion of pollen from one flower and with the egg of the other flower, other plant, is called fertilization. Okay. Next, pure line breed. So pure line, what is pure line breed or what is true breeding line? A pure breeding variety is a plant that undergoes self-pollination for many generations with the stable trait inheritance from parent to offspring and maintains homozygosity. So listen here, if a plant, for example, a plant which has the characteristic feature of a tall, okay, tall plant. So first generation, the tall parents produces only tall plants. Okay, the F1 offspring again when it happens to do the self pollination or self fertilization, again in the F2 generation also they produced only the tall plants. Yes, and then in the next generation, as like generation after generation, if they produce only the particular trait which is exhibited by their parents, means it is said to be as pure line breed. They maintains homozygosity. What is homozygosity? Now we will see. Homo means what? Same. Okay. Next, emasculation. What is emasculation? The process of removing anthers from the flower of a plant. It's said to be as emasculation. The process of removing anthers from the flower of a plant. Just see, as I said, he has selected the flower. It is such a manner, it is large in size. So easily the anthers can be removed with the help of a small scissor. This is what's said to be as emasculation. And next one is bagging. What is bagging? The process of covering the emasculated flower with a butter paper or polythene bags